In this video, I'll try my absolute best to explain the lac operon and gene expression and regulation in bacteria for 6-year-olds. Even though you may be older than 6 years old, or by almighty chance that you are younger, I hope that you learn a lot from this video. I am here to present and you are here to enjoy. The lac operon was first discovered in a bacteria known as E. coli that was designed to help the bacteria chemically break down lactose. The operon was to be commonly found in prokaryotic cells and bacteria, and it became to be known as a genetic regulatory system found in bacteria in which genes coding for functionally related proteins are clustered along the DNA. Okay, for the general structure of the operon on the DNA, there are 4 parts with an acronym that will help us greatly with remembering the parts of an operon. The acronym is PROG, which is spelled P-R-O-G. In PROG, P stands for promoter, R stands for repressor, O stands for operator, and G stands for genes. The promoter is the region or specific sequence of nucleotides on the DNA where the RNA polymerase can bind on in order to initiate transcription. The operator is the on or off switch that will activate the operon. Then the repressor is an enzyme that would inhibit the operator. Finally, the genes are the segments of DNA that the RNA polymerase rides along to create mRNA that would create proteins that would break down the lactose. Additionally, for bacteria, when there are genes that work together to complete a certain task, they are located next to each other and are controlled by the same operator and promoter. In this case of the lac operon, with the purpose of breaking down lactose, the LAC-Z, LAC-Y, and LAC-A genes are next to each other. Alright. Now that we established the parts of the operon, we will look at gene expression and regulation when lactose is present. In this graphic, there will be lots of lactose present right now. Eventually, one of the lactose will bind to an allosteric site of the repressor and change the shape of the repressor. As a result, the repressor will no longer bind to the operator. As a result, the operon will be activated. RNA polymerase will go downstream of the DNA and create mRNA from each gene to create the proteins to remove the lactose. When the lactose is gone, the repressor reverts back to its original shape and will attach back to the operator, deactivating the operator. Now the RNA polymerase cannot move past the repressor and the operator. Then, when the lactose is present again, the lac operon process starts again. Whew! Wow! That was a lot of information. Anyway, I hope you all learned a lot about the lac operon and gene expression and regulation. Thank you so much, and goodbye.